Thank you so much for the introduction. Well, good afternoon, everyone. My name is Pengfei Zhang from Arizona State University, and I'm genuinely excited to share our work with you today. So we have been exploring a foundational question in immunotherapy and vaccine design. The question is, how reliable are our models when predicting T-cell receptor and epitope binding? And what we found is something that is quite surprising, and those surprising vulnerabilities can merge, not because of the poor model design, but because of the data that those models are trained on. This project was a collaboration with Hao Mei, Sejin Ben, and Heibuk Li, and it's been supported by the NIH. So let's quickly go over the biology. T cells use their receptors, or TCRs, to recognize short peptide fragments called epitopes. And those peptides are presented on the surface of cells by MHC molecules. When a TCR binds an epitope, it triggers immune response to remove affected cells. So what does it mean for a TCR to bind to an epitope? Well, biologically, it is a precise sequence-driven interaction. And experimentally, we can validate those bindings, but in the bio, the data is very limited. You can think of the TCR as a cop trying to identify the criminals, which is epitopes. And here, studying the interaction between the TCR and the epitope has important applications, such as vaccine development or even for cancer treatment. Now let's shift into the computational side. So the goal here is pretty simple, at least on the surface. Given a TCR and epitope sequence, can we predict whether they bind or not? We frame this as a binary classification task, where we wanted to input a TCR and an epitope sequence and output a 0 or 1, indicates binding or not. Now here's the issue. When we train a binary classification models, we do need the positive and the negative data. While we have a decent amount of positive binding pairs collected from different databases like IDB or VDDB, we have very few validated non-binding pairs. That means to train those models, users typically have to generate their own negatives, and the field kind of relied on two main heuristics. One is called negative shuffle, the other is called negative healthy. The idea of negative shuffle is basically we wanted to shuffle the TCR and epitope sequence from the already existed databases. And the assumption is that a TCR binds to epitope A is unlikely to bind to epitope B. And the negative healthy means users would just randomly sample TCRs from healthy repertoires and pair them with disease-related epitopes. And the assumption here is, the healthy TCRs don't recognize diseased peptides. While those strategies are widely used, they're kind of based on the assumptions and can introduce noise or bias to the data. And meanwhile, most of prior work has focused on other aspects like model design. They're trying to experimenting with different architectures like convolutional neural network, LSTM, transformers. Or they're trying with various embedding methods like static embedding, blossom metric, or context-aware embedding, CAT-ALMO. But very few have asked, what's the impact of those negative data? And how diverse or realistic is our training data? And that's where our work comes in. So the motivation of the project came from a generation task where we train a TCR sequence generator using reinforcement learning. So unlike the prediction model where we use epitope and a TCR as input and predict the binding probability, over here, we supervise fine-tune a protein language model to generate TCR sequences conditional on the given epitopes. And then the evaluated sequence will also be evaluated through a prediction model. And to further encourage the generative model to generate more sequence with higher binding affinity score, we use the reinforcement learning framework. And here, the prediction score is used as the reward signal. Specifically, we're using the PPO to train a policy that learns to produce those TCRs that can maximize the evaluation score. And during the training, we use a technique, we use the logics instead of the probability because the logics can provide a fine grained gradients compared to the threshold probability. So here's an example from the training log where we're given the input epitope to the model, and then the model would start to generate the TCRs with certain reward scores, the binding scores. At first, the generated sequence looked okay, but after a few optimization steps, something strange happened. So as you can see, the BP score kept it going up, even when the sequence became completely unrealistic. Sequences like just A or AWT were scored as very strong binders, with BP logics as high as 38. Now, this is clearly a failure case. The model is assigning a very high confidence to sequences that are biologically implausible. This is a classic case of reward hacking in reinforcement learning, also known as hallucination if you're using ChatGPT a lot. So the model finds a loophole, a way to get a high reward without actually solving the intended task. In our case, we call this as a BAP attack because it exposes how our binding affinity predictor can be tricked into assigning high confidence to invalid inputs. Now, what's fascinating is that this process actually became a diagnostic tool. And we can now intentionally attack those prediction models to find their blind spot, and then use those blind spots to defend and retrain them to improve the models themselves. So we propose this BAP attack and defend framework. Specifically, in this framework, we first attack those reward model. In our case, it's the binding affinity prediction model, and trying to find the weakness of the model, and then improve them. And the first step, we supervise fine-tune a protein language model on those epitope and TCR pairs. The goal is to generate those TCRs then conditional on epitope sequence. Once the sequence generated, we evaluate the TCR sequence's quality through a binding affinity prediction model. And we use the logics from the model as a reward signal to further update the LLM policy. Once the training is finished, we'll have access to all the generated TCR sequences. Now, we do need to understand how much of those TCR sequences are false positives, how much of them are actually the limitation of the model. So the next step is to identify those false positives. To do that, we utilize two extra models, a TCR match and GPT-LL, to measure the authenticity score of the sequence. The TCR match is a keyword-based method, and the GPT-LL is a log likelihood-based method from the large language model. We define a sequence as false positive if this sequence has a very high binding affinity score, but with a very low authenticity score. Now, once we identify all those false positive pairs, we call those dataset as adversarial negative dataset, and we sample additional 3 groups from the reserved pool dataset we had before with certain ratios. So in total, this new dataset have all 4 different groups with certain ratio. And we're putting all those data sets together, try to fine tune the base model to get the updated model, M prime. And then we repeat this attack and defend framework until the model, the framework could no longer find significant amount of false positive sequences. So with this framework, we first wanted to show that, is this a BEP attack just a problem of our prediction model, or is it more of a universal problem for all the prediction models out there? And we show that it's not just a problem of our prediction model. So here we attack 5 different BEP models with 1 iteration. Those 5 models were widely used in the community, and we picked those models because they cover a diverse range of architectures or embedding method. And on the right side, we show that the x-axis is the training steps, and the y-axis is how many false positive counts we could find. We show that as the steps go on, for each of those models, we could keep finding more and more false positive counts. Although there are some models can be easily attacked, some models takes extra steps to be attacked, but we show that all those models can be attacked. Although we only attacked 5 different models, we're pretty confident that this BEP attack is a universal problem because all those models are approximate model, and they were trained on the data set, and model can make mistakes. So now the next question we ask is, can this attack and defend framework help? So for each of the prediction models, we try to attack and defend them for 3 iterations. And before we show the results, I want to emphasize that there are 2 questions we really care about. The first is, as iterations go on, are we observing more or fewer false positives? And second is the performance score for each group increasing or decreasing? So we attack all those 5 different models for 3 iterations. On the left side, figure A, we show that as iteration continues, the false positive counts dropped dramatically from thousands to less than 100. And in terms of the model performance for each data group, in B, C, D, E, model performs significantly better for the adversary negative group, jumping from 0 to more than 90% accuracy, and without jeopardizing the performance of other 3 groups of the real data. In order to better understand those data, we also did some visualization analysis. Here, I show the Disney plot for the false positive samples we identified from each prediction model in iterations. Each data point is embedded as representation vector first. What we observe is that as iteration continues from left to right, the number of false positives clearly decreases. And more interestingly, the clusters became less distinct and more entangled. This suggests that as the models became more robust, they're less easily fooled by specific false positive patterns, and adversary sequences became more diverse and harder to isolate. Additionally, we also show the comparison of the sequence themselves with the seq logo. So all sequences were center-aligned using a special panning token X to make the same length. And by the way, this panning technique is widely used to compare TCRs in the community. And we can clearly see that at the initial iteration, each prediction model has very different and very model-specific limitations. And they're very easy to support. But later on, as the model became more robust, the sequences looked more similar to each other, became more model agnostic, and more difficult to support. This further confirms that the models are becoming more robust and consistent in rejecting unrealistic sequences. And once we have collected all those false positives from different models and different iteration, we start to put together a combined negative data set and hope that users in the community can directly use those negative data set to improve their model without going through any attack and defend process. So to begin, we evaluate the robustness of the 5 existing models against adversary generated negative examples. As you can see here, for the first 2 bars for each model in orange and purple, the base model, even when supplemented with simple heuristics, struggle significantly. Some models misclassify over 80% of those adversary sequences. However, the green bars show the performance after fine-tuning with our proposed attacking and defend framework. This improvement is dramatic. Across the board, our method boosting the true negative rate to over 90% for most models, with PITE and Cadamo MLP reaching nearly 99%. And the key takeaway here is that our framework effectively teaches the model to recognize and reject those biologically implausible sequences that were previously blind to. And a critical question, of course, is whether this new robustness came as a cost of performance on standard authenticity data. We show improvement on the adversary negative data set, but what about the performance on other real the groups on real data set? So if our method was simply overfitting to the adversary example, we would expect to see a drop in performance here on the real data set. Instead, we see that the fine-tuned models, the green bar, achieve performance that is comparable to or even slightly better than the base model across all the 3 standard databases. Now, this is a crucial result. It demonstrates that our framework is not a simple trade-off. We can significantly enhance a model's robustness against sophisticated false positives without sacrificing the performance on the real data. To conclude, I'd like to leave you with 4 main takeaways from our work. We first highlight a critical issue. So even the high-performing binding prediction models can be fooled by biologically implausible sequences. And through adversary generation, we systematically expose those failure cases. And by retraining on those examples, we significantly improve the model's ability to detect the false positives without sacrificing the performance on real data. This framework generalizes across multiple architectures and embedding methods, showing that this is a model-agnostic vulnerability. It also produces a reusable adversary benchmarking dataset, which can help evaluate future models for robustness. In short, this work shows that robustness matters and that we can proactively improve it by identifying and fixing blind spots through controlled adversary attacks. Ultimately, our work provides both a framework and a resource to build more reliable and trustworthy prediction models, paving the way for safer and more effective immunotherapies. Thank you guys for your attention.
Thank you very much, Pengfei. We have time for 1 or 2 questions.
Thank you for your talk. It was very interesting. I was a little confused by your example, and I'm wondering if this is just a toy example, but the epitope for a T cell receptor isn't just a peptide, right? It's a peptide MHC complex.
That's correct.
Then your generated TCR, was this the CDR3 beta sequence? And I'm just wondering, and then when you showed the false positives, were those just non-plausible protein sequences? Did it have anything to do with whether these were true binders? And how did you validate whether, I guess I have a lot of questions. How did you validate that these were binders? And how could you predict the binding of a TCR to an epitope without knowing the MHC, just with the peptide? And those are all my questions.
Great. Those are great questions. So first off, about the MHC part. In this work, we're focusing on the binding between the epitope and CDR3 beta of the TCR specifically. That's why we're not considering the MHC in this work. And for your second question about how do we validate whether this is a true binder or not when we generate those sequences. So what we did is, so the best way to validate those binders would be using the wet lab evaluation, but that is very expensive and is not applicable in our lab. So what we do is we're using a state-of-art approximated prediction model that is trained on a large amount of data set. And we're using this prediction model to give us a kind of a weak supervision or the weak signals of whether they're binding or not. And to identify like whether those are the false positives or not, we're using the authenticity scores from another two models, in our case, TCR match and GPT-LL. And about those sequences, so when we generate those false positive sequences, many of those sequences, I would say probably they are not even a protein sequence. They're really just showing the limitation of the prediction model. So over here, we cannot tell whether this is actual protein fragments or not, because the goal of this project is to learn the limitation of the prediction model and try to improve that limitation. So we're going to take all the limitations we learned from the generative model, and then using those kind of, I guess in the LLM they call this hallucination. So we're trying to using those hallucination knowledge to further refine the model and making the model more robust. Did that make sense? I hope it answers the question. Any other questions? All right. Thank you guys again for your attention.